Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study that could revolutionize how we diagnose and treat kidney disease in our canine companions. This research, recently published in the Journal of Veterinary Internal Medicine, focuses on the role of microRNAs, specifically microRNA 126, in dogs with immune complex-mediated glomerulonephritis, ICGN. Kidney disease is a significant health concern for dogs, often leading to illness and, sadly, death. Glomerular diseases, such as amyloidosis, glomerulosclerosis, and immune complex-mediated glomerulonephritis, are major culprits behind chronic kidney disease. Diagnosing these specific glomerular diseases can be challenging. Currently, a definitive diagnosis requires a renal biopsy, an invasive procedure. That's right. A renal biopsy involves taking a small tissue sample from the kidney for examination under a microscope. While it provides valuable information, it's not without risks. Some dogs may not be suitable candidates for anesthesia or the procedure itself. Exactly. So, the quest for a non-invasive diagnostic tool has been a priority in veterinary medicine. This is where microRNAs come into play. MicroRNAs are small molecules that regulate gene expression after transcription. Think of them as tiny switches that can turn genes on or off. They're involved in various biological processes, both in health and disease. What's exciting is that these microRNAs can be found in body fluids like serum and urine, making them accessible for diagnostic purposes. The researchers in this study hypothesized that the pattern of microRNA expression in dogs would be different depending on the type of glomerular disease they had. They also thought that these patterns might change as the disease progressed. To test this, they examined archived serum and urine samples from dogs with confirmed glomerular diseases, as well as samples from healthy dogs. This was a retrospective study, meaning they looked at samples collected in the past. Specifically, samples submitted to the International Veterinary Renal Pathology Service between 2008 and 2016 were used. The study included 18 dogs with glomerular disease and 6 healthy dogs for initial RNA sequencing. Then they expanded the study using quantitative reverse transcription PCR to include a larger group of 49 dogs with glomerular disease and 13 healthy dogs. The researchers used advanced techniques like RNA sequencing to profile microRNAs in the samples. This allowed them to identify microRNAs that were differentially expressed, meaning their levels were significantly different between the diseased and healthy dogs. When looking at the samples, the dogs with chronic kidney disease were divided into stages A and B based on kidney function and the severity of damage seen on biopsy. Stage A represented earlier disease, while Stage B indicated more advanced disease. So what did they find? Interestingly, when comparing more advanced disease with earlier disease, there were no microRNAs that were expressed differently in serum. However, in urine, microRNA 21 and microRNA 182 were higher in dogs with more advanced disease. This suggests that these microRNAs could be markers of disease severity and the presence of tubulointerstitial fibrosis, a type of kidney damage. But the most striking finding was related to microRNA 126. Yes, the expression of microRNA 126 in urine was significantly higher in dogs with immune complex-mediated glomerulonephritis compared to dogs with glomerulosclerosis, amyloidosis, and healthy controls. In fact, it was more than 10-fold higher compared to glomerulosclerosis, nearly 30-fold higher than amyloidosis, and over 100-fold higher than healthy dogs. This is huge. This suggests that microRNA 126 could be a specific marker for ICGN. To confirm this, the researchers used quantitative reverse transcription PCR, a more targeted technique, on a larger set of samples. And the results held up. The PCR confirmed that urinary microRNA 126 levels were much higher in the dogs with ICGN. They then performed what's called a receiver operating characteristic curve analysis to assess how well microRNA 126 could distinguish between ICGN and other types of glomerular disease. The area under the curve was 0.95, with a sensitivity of 90% and a specificity of 92%. This means that microRNA 126 could correctly identify 90% of dogs with ICGN and correctly rule out ICGN in 92% of dogs without the disease. Those are excellent numbers. This level of accuracy is really exciting for the potential use of microRNA 126 as a diagnostic tool. So, what does all this mean for our canine patients? Well, Imagine a dog presenting with signs of kidney disease. Instead of automatically resorting to an invasive biopsy, we could measure microRNA 126 levels in the urine. If the levels are high, it would strongly suggest ICGN. And why is that important? Because ICGN is often treated with immunosuppressive drugs. However, these drugs can have side effects and are not appropriate for other types of glomerular disease. Therefore, knowing whether a dog has ICGN is crucial for guiding treatment decisions. So, microRNA 126 could help us identify dogs that would benefit from immunosuppressive therapy without needing a biopsy. Exactly. It could also help us avoid unnecessary immunosuppression in dogs with other types of glomerular disease. The study also shed light on microRNA 21 and microRNA 182. Yes, these microRNAs appear to be associated with disease, severity, and fibrosis. As kidney disease progresses, their levels in urine increase. This suggests they could be potential markers for monitoring disease progression and assessing the effectiveness of treatment. The researchers also explored the roles of microRNAs 335, 128, and 486. Their data suggests that microRNA 335 and 128 could be biomarkers for glomerulosclerosis and amyloidosis, respectively. Also, microRNA 486 may play a role in the progression of chronic kidney disease. Of course, this study has some limitations. The study focused on the 3 most common types of glomerular disease. It didn't include other, rarer types. So, we don't know if these findings apply to those conditions. Also, because it was a retrospective study, there were some variations in how the samples were collected and stored. This could have affected the results. Despite these limitations, this study represents a significant step forward in our understanding of kidney disease in dogs. The discovery of microRNA 126 as a potential non-invasive biomarker for ICGN is particularly exciting. It could revolutionize how we diagnose and treat this common condition. Further research is needed to validate these findings and explore the potential of microRNA 126 in clinical practice. But the future looks bright for dogs with kidney disease. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.